Right, guys, let's get started. So good afternoon, everyone. I'm Mohammed Alser. I will be talking about different topics other than all the previous lectures. Professor Anur is busy presenting some work in a conference, so I'll be giving the lecture for today. And this is my email. If you feel interested in some of the topics I will cover today, so please feel free to contact me. I will be happy to work with some of you on some of these topics for my research as well as other members' projects. So I'll be talking about accelerating genome analysis. System design for bioinformatics is a critical problem. Why is that? Because it has a large scientific and medical implication as well. This lecture is about accelerating a key step in bioinformatics, which we are focusing on genome analysis, and in particular, read mapping process. There are many other bottlenecks exist due to the movement of data because genomic data is really huge. So moving this data from the hard drive to the processor, processing it at that side causes you some latency. So even if you have some accelerator, GPU or FPGA or other processors, you still face the same problem because initially the data is huge. We will cover various recent ideas to accelerate this read mapping process as well as genome analysis in general, and most of those ideas are our own, which we developed in Safari Research Group. The recommended reading for today, I recommend you to have a look at those two books. They are not papers, so you need some time to go through them. You may find some outdated information, but still most of the algorithms are related and applicable even today. And basically, those two books should give you the base knowledge for bioinformatics as well as genome analysis. I'm not going to explain to you how to process the biological data using those tools. The goal of this lecture is how to accelerate those processes. All right, so I'll start describing the problem we have, which is basically DNA read mapping. And I will show you the state-of-the-art read mappers. And then we will show some algorithmic improvement that we can achieve to accelerate this process, as well as hardware acceleration. And finally, I will go through the future directions that we are targeting currently in our research group. All right, so our life begins with a cell. And in a human body, we have billions of those cells. Those cells different in size, as well as the amount of them. But they are common in the content of those cells. So all cells have a DNA, or 23 chromosomes. And those 23 pairs of chromosomes are unique in all, are common or same in all human body cells. So how the liver is different from, for example, the heart, from your skin, eyes, and other cells as well, if all of them have the same content or genetic information. So a cell is basically the smallest structural unit of our organism that is capable of independent function. So the cells store all your genetic information. Whatever you need to know about your genotypes or phenotypes, you will find it in your cell. So all cells have, again, the same information. So if you read the genetic content of single cell, then you are done. You already know everything about the human body. You don't need to go through cell by cell. So again, the question, how the heart cells, for example, different from the liver, the kidney, or other organs of the human body. So they all differ in the genes, the expression of the genes. So you have the same amount, exactly the same amount of genes, the same genes in all cells. So you can think about it as you have 20,000 or 25,000 switches in each DNA. And if you switch some of them on, some of them off, you will have different function, or different cell, or different even organ. So this is how your cell is different from other cells, but all of them have the same content. So either activating some of them or turning them off for some time. So you could turn them on after some time. So that's why the diseases may appear after 10 years, 20 years, though you already have the genes of that disease since beginning. All right, so initially our life depends on 3 critical chemical molecules. Those molecules are DNA. We replicate it to start reading the DNA. And in the next slide, I'm going to explain what is the DNA. And you can think of the DNA as a cookbook where you have many recipes. Then you transcribe the DNA into RNA, which is something unstable that could be converted into a protein. So your DNA is stable, cannot be converted directly into protein. The RNA, you can think of it as a recipe, single recipe, or many recipes that have the instructions to do something. Then you translate this recipe or the set of instructions into something visible and could be invisible as well, which we call it a protein that codes you the phenotype. Phenotype is something like hair color, eye color, and so on. So this is basically your meal after you perform the recipe. All right, so you can see clearly that everything depends on your DNA. So if your DNA is different, obviously those steps will be different. So now let's go through the chemical structure of DNA. Basically, the DNA is built up of those 4 main chemical molecules: adenine, thymine, guanine, and cytosine. So adenine and thymine are linked together with 2 hydrogen bonds, and guanine, cytosine with 3 hydrogen bonds. In this way, they are stable, chemically stable. They cannot have chemical reaction with other components. So they are linked together in a double helix form. And you can see here, for example, you have all the content of those chemical molecules. So the C, as we explain here, is always linked with the G, which is guanine. Cannot linked with the T because won't be stable. So A is linked with T, and so on. So you can see that one strand of those double helix structure is enough to read the content of the DNA because the other strand will be the complement of the first strand, right? So if you already know at this position A, then the other strand has definitely T, right? So our goal is to read this sequence, which is ACGTs. So if you try to stretch the DNA out of the cell, then it will be 2 to 3 meters long. So imagine if you have billions of those cells and your DNA is stretched out as a single segment. So you can imagine your body shape. That's why the DNA is super coiled. So it has a coil, and other coils. So it is super coiled such that it saves the space. So it can fit inside a cell. A cell diameter is 4 to 100 microns. So you can imagine without the supercoiled structure, how it looks like. All right, so now we know the structure of the DNA. So how long is this DNA? How many characters, how many ACGTs we have in a human being, for example? All right, so here we have a virus. We call it 5X174. In this virus, we have 5,386 characters in the DNA of this virus. This virus infects the E. coli bacteria. This bacteria coming from undercooked food or unwashed vegetables and fruits. So this bacteria DNA has 5.4, almost 5.5 million characters, ACGTs. Again, all DNA has only A, C, G, and T. The human body has about 3.2 billion characters. An onion DNA has 16 billion characters. You can imagine almost 5 times the content of the human DNA. This is very rare flower, this Japanese flower, the DNA has almost 150 billion characters. So you can imagine we are not the superior here. All right, any guess what's this?
It's a transcription of the human DNA.
All right, excellent. Yeah, this in UK in a museum, they print the human genome entirely, everything. They build it as a series of books. So this is how it looks like. We have 23 pairs of chromosomes. So you can see, for example, chromosome 14 could fit into 4 books. So the content here, only ACGTs. You can imagine part of the text here could affect your eye color, your hair color, or cause you some diseases or other phenotypes. So this is the first printed human genome, 3.2 billion characters printed with a font size of 5, weigh 450 kilograms. So you can imagine how many of those DNA you hold day by day.
So is that like the average of a lot of humans, or is it just like one human?
No, this is the reference genome. I'm going to talk about it in the next couple of slides.
Is this now just the one strand or the
This is one strand.
Okay, so actually it's double the amount of letters.
No, basically this is a single one. So they read one of those double helix structure, and the other one is complement, basically.
Oh, yeah.
So you already, if this is A, then it's linked to T, and so on. So normally we are always interested in this sequence. The complement is just complement. So if you know this, you know that, right? All right. This is, again, the 23 pairs. So we are always reading one of them, this side, for example, not the other side, because, again, it's complement. So, again, this is your 23 pairs of chromosomes under a microscope. This is chromosome number 12 of Henrietta Lacks. Does anyone know who's Henrietta Lacks?
The cells that are immortal or something?
Yeah, excellent. All right. This is the lady. She had a cancer, and she undergone a surgery. And the doctor extracted the tumor, and he kept it for some time in a safe place, and then distributed to all the research labs all over the world. So whoever doing cancer research is basically need to do something with those cells. So this lady saved almost the world by contributing her cells without her awareness even. She didn't sign any legal documents, anything. Even the doctor did not consult with her at that time. And basically there are many research these days try to reveal the genetic content of her relatives. So now they know, for example, the diseases that her uncle has, for example. So you can imagine that. By reading the DNA, because the similarity between a person and other person is 99.9. So if I know, for example, anyone's DNA, then I could infer the DNA of his brother, because there is 99.9% similarity. And this is an actress. They did a movie about her story, so you can watch it. You get more information about this story. So again, this is the reference genome that they printed in London. So in this reference genome, it's a collaborative work between US, UK, Japan, and other countries. It took them 13 years to read the content of the human genome. You can imagine 13 years just to read the content. And it cost them $3 billion. So it's 3.2 billion characters. It cost them $3 billion. So you can imagine each character cost them $1. So in this reference genome, basically all the research on a human being is built upon this reference. So whatever you do in research, you basically rely on this reference genome, which is constructed fully in 2003. All right, so if you read the content of somebody's genome, what's the importance of that? Why do you need to do such process if it costs you too much money and takes of your time? One of the applications, for example, if you have a database where you store the content of the DNA, and then you find in a crime scene, for example, sample DNA, then you want to recognize the owner of that sample. So you could look up in this database and find a similar owner or the owner himself. So this is one of the applications. The other application, if I have a sequence and I have many other sequences, I want to know the similarity and the differences between those bunch of sequences. Why is that? Because I want to recognize, if I know this guy has such disease and because of that gene, for example, at this location, then I could analyze my sequence as well and check if the same location have the same gene as that guy, then possibly you have already that disease or you will have it in the future. So it's not necessarily to have it in the time being. Maybe your kids will have it in the future. You don't know. The third application, for example, if you have a sample and you don't know whether that sample is a bacteria or virus. So you could build up what we call a phylogenetic tree where you cluster the similarity between many other species and you see if it is similar to this guy in bacteria or virus or other organism, then yeah, you could, and probabilistically, you could say that this guy, hey, yeah, he's similar to virus, so you may get infected with a virus, for example, something like that. And definitely you want this process to be extremely fast. It will be like useless to perform this, for example, in a month. So you're already infected by virus or bacteria. Why I would know after a month what's the cause for that? So this needs to be performed a couple of minutes or even a couple of seconds. This is our goal. Again, the similarity between species with the same organism typically is really high. So human to human, 99.9%. And again, if he's your relative, then the similarity is much higher than that. And with other organisms, you still have you sharing part of your genomes with other organisms, even with a banana. So you can imagine that. All right, so our goal to know the content of our genome. Or could be a yeast, could be a plant, could be a flower, could be anything, right? Again, our goal is to reveal the content, the entire content of our sequence, A, C, G, Ts. And basically, once we've done that, we don't need to do a wet lab setup. So we don't need to interact with those chemical molecules in the lab. Once you get the sequence, then it's a computer science problem. And I'm going to show that. All right, how we will read this content? Imagine you have a black box. It could be a machine or technology or a method even. We don't know what is it so far. And I want to give it a picture. I want to capture the content of that picture as it is without any changes. So this will be a chemical molecule, and I want to read it in a human-readable format. Unfortunately, we don't have such a machine or technology or a method that you give it a DNA or genetic sample and give you the content as it is. What we have is this. So this is the bioinformatician, and try to figure out the content of this vase. So we have a machine that breaks down the original picture into very small pieces. It could be small or long as well. So the first sequencing technology, sequencing because we want to read the entire sequence. So we call it sequencing technology. This one we call it second sequencing technology, and give you very short pieces of the original picture or the original genome, for example. So it reads only small, very small content of your genome. The other technology, we call it third generation sequencing technology, which give you larger pieces. And now the problem is to link those pieces together to build up the original picture. So you already know the content of each piece, which is ACGTs in our case, and we want to link them up together to build up the entire sequence. So it's like a loop. You already have the entire content, you break it down randomly, you don't know which is linked to which, and then you need to build it again. So what do you think? Which one is better? To have small pieces or large pieces?
Large pieces.
Okay, yeah, theoretically, you need to have large pieces. It's easy to match or to link them together, right? Unfortunately, so far, we could generate very short pieces, but the machine is very accurate. So it will give you the exact content with very small error probability of the content of each piece. However, for the larger pieces, we have very high error rate. So the sequencer machine generates this much of error. So imagine if this piece is, for example, 10,000 characters long, and you have 15% of them wrong. So you cannot infer what exact character caused that disease, for example. Because it could be genetic variation, it could be a sequencing error. So that's why both of those technologies are still in use. So these are examples of the sequencing machines, and could vary in size, fridge size, for example, all the way to an iPhone gadget. And all of them are in use. Okay, how a sequencer read your DNA. So basically, as I said, we cannot read the entire sequence of the DNA. So what the sequencer does is chopping your DNA randomly into very small pieces or long again, and pick one of those pieces on a glass surface, and based on the chemical reaction between the C and G, T and A, it's doing reverse engineering and try to, for example, add a T molecule to this segment near the surface. So T should theoretically react with the A, right, to form a stable chemical molecule, right, with two hydrogen bonds. So if this reaction happens, then you already know that the content was A, right, because we add T. And you keep adding all the 4 molecules to each character in this segment near the surface. So once the reaction happened, you observe it by optical sensor, and you produce the output to the user, which is A. Then you wash the surface, and you add other molecules. You continue on. So C should react with the G. Then if this reaction happens, you observe it by optical sensor. You produce the output. However, the sequencer doesn't do this one by one. That's why we call it high throughput sequencing. It is done basically million of them, or depends on the sequencer, could be thousands or millions of them in one time. So here you have randomly those segments that chopped from the original DNA. And this is how the optical sensor looks like. Every time we add the molecules, you observe the reaction, and you recognize the reaction based on the energy, based on the light produced, based on many other properties. Depends on the sequencing technology we use. And then the sequencer will give you billions of those in 1 second, for example, or a couple of days, couple of hours, depends on, again, on the sequencing technology. Now your problem becomes worse. So originally you have the original DNA, and now you have billions of millions of those. So what to do? And the worst problem is that those segments do not have any information about the order or the location in the original genome. So you have basically a bunch of those characters, and you don't know whether this is linked to that or which one comes first. So what you need to do is to link them again using some algorithms, some technology, some devices. And after that, you do the subsequence processes, like analyzing the DNA, compare it to other references, and then for those genetic or phenotypes. All right, so our goal now, this is your input, which is short reads or long reads. We call them read, which is the short segment produced by the sequencer. We want to take those as input, and we want to produce the entire sequence of your DNA. So the main question here, before start building your genome, you want to ask yourself, do you know the species of the subject sample you are targeting? If you know that I'm having a sample of bacteria, then it's much easier than you don't know. So if the answer no, so what you need to do basically is to link those reads based on the similarity with the first character of the segment and the last character of the previous segment. So those nodes basically are your reads, and you link them based on single character, and you build what we call a De Bruijn graph. So the edges, if you have an edge between a node and another node, means those reads should link to each other. However, because we have millions of those reads, so you will have a very messy graph because we have a large number of nodes and edges. And what we want is just horizontal, nicely written sequence of those ACGTs. So there are many heuristics to clean up this graph and remove the loops. Otherwise, your graph will look something like that. So how to infer the sequence out of this graph. Again, if you know the species that you are targeting, which is much easier step, then imagine you have this Switzerland flag, for example. So it could be as a puzzle where you have those large pieces, and you want to connect them together because you already know the original picture. So if you have those small pieces, even if they are like randomly scattered or chopped, you still are able to build them again because you have the reference. Same thing if you have very small pieces. It will be harder step, but it's something doable. You can do it. And this is what we are targeting. So if you already have a reference, which is your picture to solve the puzzle, you have your sequencer here generating those billions of reads or segments, and you want to link them back to your reference. So you have the reference, which is the picture in the puzzle, and you have those puzzle pieces. You want to link them together based on the similarity. So if this picture is similar to this portion of the reference, then yeah, it's better to put it here. And so on, you keep doing this with all those small pieces. After that, you have the entire sequence of the subject, not the reference, because we already used the reference to build those, to link those reads. So you get this entire sequence, and after that, you start like inferring or applying your studies or whatever you're trying to do here. Could be personalized medicine as well. All right, so we already explained the sequencing step. Now the second step in genome analysis is read mapping, where you try to link those short reads to your reference genome. And read mapping, we define it as you have many short DNA segments, could be short or long again, and we want to map them to a known reference. So this is the condition here. You need to have a reference for that species, and you need to map this segment to that reference. However, you need to allow for some differences. Why is that?
Because the reference genome isn't exactly the same as the genome we're looking at now.
Yeah, because we are targeting to infer, for example, the genetic variation. So if we don't allow for this variation, we cannot get any benefits, right? We already hide those those variation. So we need to allow for those while we are mapping. So it's not exactly similar to a puzzle problem, because in puzzle problem, you look exact match between each piece and the reference, right? So here's the challenge. So basically, this is what we need to do. You have the DNA, you already read it into short pieces, and then you keep matching piece by piece to the reference genome. For sure, you will allow for some differences. This is a threshold that is defined by a user normally. So if you are targeting between a human and a human, then the similarity is really high. Then you don't need to allow for too many differences, for example. And after you map the reads to a reference, then you got your full sequence. Yeah?
That means that the reference genome must be random enough that you don't get too many matches. Like with your short reads and so many characters, you can have possibly many matches. Exactly.
Imagine if your read is 100 character long, and you reference 3 billion. So definitely each read will match for maybe thousands of times in the reference. So this is another challenge, basically. So it is challenging to map to a human reference rather than a mapping to bacteria, for example, because it's very short, few thousands, not a few millions. So the reference genome is really hard to build again. So we have for each country basically has its own project to sequence the citizens of that country. So for example, US, they have 1,000 genome project, they have millionth genome project, Japan, same thing. Turkey started recently. I'm not sure about Switzerland, but most of the countries spending billions of dollars on sequencing the citizen of the country. Because as you know, if you live, for example, you have a twin brother, and each one of you live in different country for a certain time, for example, 20 years, then again, you read the content of the DNA will be different than before 20 years. So you can imagine that. So the way you live your life affects your DNA as well. So that's why each country should sequence the content of the DNA rather than just relying on a single sequence for the entire world. So again, mapping those pieces to the reference is a challenging step because we need to allow differences, as well as each segment could map to many locations in the reference genome. So those are the challenges that I just mentioned. So we could end up having many possible locations in the reference. We need to tolerate many variation, many errors as well, because the sequencer has its own error, and need to map each read very fast. Why is that? I have the example of, for example, critically ill man, and he needs to know the disease or the bacteria or virus, so it's useless to have it after a month or a year, for example. So you need to have it in a couple of seconds, couple of minutes. All right, so we do this using two styles. So either we have this read mapper is the algorithm or the hardware that perform this step, which is matching or mapping your short segments to a reference genome. So it could be either using hash-based read mapper or Pearl-Wheeler transform. Using the hash table, how many of you know the hash table? Okay, great. In a hash table, you could think about it as two-dimensional table where you have in the first column, you have short words, and the second column could be the location of those words. So you hash or you index some very long string, and you basically select, for example, the word or the seed with 10 characters. Then you have like 10 characters, and the location of that 10 characters in your entire string. I'll show you examples in the next slides as well. So we use this hash table to basically to query or to check if this small word or small k-mer exists in that reference or not. And because we have a lot of repetition in the reference genome, then use hash table makes sense, right? Because it can help you to reduce the size rather than recording single word many times in the reference genome. You record it once, but you record all the location that happened in the reference genome. And because we record everything, all the location, this is very sensitive. Sensitive means very accurate. We'll give you all the mapping location because we record basically everything, right? However, it is slow because we need to go location by location in the reference genome and check the similarity with the read. Obviously, it requires a large memory because essentially the reference is really large. And when you try to index everything there, and you end up having very large hash table, definitely it will be less than the size of the reference genome. It could be somehow similar to the same size if depends on the way you select those words. If you select, for example, non-overlapping spaced seeds, spaced mean you select this word, you want to index it, then you, after 10 characters, you select another word, for example. So you save space, but you lose accuracy because you don't check every position in the reference genome. The other method is you can think about it as a tree-based. So yeah, sorry, I forgot to mention one important thing. In hash table, normally if you want to check something exists or not, you always need to look for exact match. You cannot check differences, single difference in the word. For example, if you are looking, for example, Switzerland, if you change it to Netherlands, you still, if you want to check the hash table again, then you go through different entries and check if it exists or not. But in tree shape, you have the node, which is the root. Then you go through the branches. If the first, for example, 5 characters are common between the 2 words, then you go through the same branch. And if the last character is different, then you end up having 2 branches only and the last character. So you save size by this method. In a hash table, you need to record everything. If this word is different than the other word by single character, then you have 2 entries in the hash table. Do you get the difference between those 2 methods? So that's why in the second method, if they want to speed up the process, they don't allow for traversing through the entire tree, for example. Otherwise, you keep visiting all the branches of the tree. Oops. By limiting the number of differences between the 2 segments, then you are reducing the sensitivity of your read mapper. So you don't allow to check all the differences, for example. Otherwise, you will keep, again, visiting all the branches of the tree. That's why we are focusing in this talk about hash-based read mappers. So in hash-based read mappers, we have 2 key steps. The first step is to do a pre-processing, which is basically building this hash table. And the second step is to check if each read exists in that hash table or not. So here's an example how to build the hash table. And because you are aware of the hash table, so basically we select part of the read to be a word, and then we store it in the hash table, and we store all the location that this word exists in the reference genome. So at location 12, mean the character number 12, we have this word starting from this character. And 35, we have again the same word, and so on. So you can imagine if this is very short, then the location list will be huge. And if this is very long, we will reduce the location list, but we will have a very long word list. Why is that? Because you reduce the possibility to enumerate all of them. So imagine if it's very long word, then it's very most probably to be infrequently happened in the same reference genome. Otherwise, if it is short, then it will frequently happening everywhere. Imagine if it is 3 characters, for example. So there is a trade-off between selecting those seeds, basically, and the location in the hash table. And there is a paper published by Professor Onur Mutlu and other researchers So tell you exactly how to select the seeds to achieve the highest performance using the hash table. So basically we want our seeds to be non-overlapping and as much as infrequent as possible. Now after building the hash table, we need to check if each read exists in that hash table or not. So this is your read. We will chop it into k-mers. K-mers is seeds or short words, and the k is the length of this seed. So if this is 12, for example, then this is 12 mers, something like that. So we chop it here into 3 pieces. Because our hash table, when we build it, we select the word size to be 12. So we chop it to the same length of our hash table. And now it's the time to check if each k-mer or each seed exists in that hash table. So you check the green guy using the hash table, you get this location list, and then you check the red, then you check the blue. And once you check, you get the location list in the reference genome. So you pick the 12, starting position is 12, so all the way to 100 characters. Why is that? Because the read length, for example, is 100. So you pick the same length from the reference, and then you compare this segment from the reference with your read. So this is your read, this is your reference, both of them same length. Then you apply some algorithm to check the similarity between them character by character. So you go all the way towards the end, then you see they match, they are same, exactly same, then yeah, this is correct, then proceed to the next location. You go to the starting location, 324, you pick the same length of the read, for example 100 with the reference, then you compare it to your read, and so on. You keep repeating the same steps. So this is different reference segment. You have matches all the way up to here, then after that all of them almost mismatched. So probably based on your threshold, if you define I want 2 differences or 3 differences, this is how you control whether you keep this segment or you just ignore it or eliminate it. So this is very high dissimilar, then just ignore it and proceed to the next locations and so on until you finish the entire hash table. Then for each read, you record the location that matched to the reference, and then you start build up or linked all those reads together. Any questions so far?
Like when you're creating the seeds, are they just like offset by 1 character, or
yeah, this is, really will affect your performance or the performance of the read mapper. And the paper I mentioned just 1 slide earlier suggests to have non-overlapping. So basically could be next to each other, could be spaced. But the criteria here is to select them as infrequent as possible. So we don't want the location list to be huge. Why is that? Because we always check at each location. So imagine if this is 1,000 locations are recorded only in this list, then we are performing this similarity measurement 1,000 times. And could potentially some of them match, so we keep them, we like them, it's good results. And some other location does not match. So we perform many computation there unnecessarily.
Do you cover the whole read sequence with k-mers, or do you only space it?
Okay. As I mentioned in the hash table, we are only allowed to use exact match query. Meaning that if you want to check if this k-mer exists or not, then you need to have it exactly as it is in the hash table. So by this method, if we cover everything, then we cannot allow for differences, right? Because you're always looking for exact match. So it's not a good choice to index everything, and it's not good to have single k-mer, because always we are looking for few mismatches or few differences. So if this is 100 long or 10,000 long, and we check the similarity by just small portion of it, doesn't make sense, right? Definitely if you have one difference or two differences will have the same impact, right? We don't want that, basically.
Maybe it's just because this is an example, but can't we actually understand that this read most probably matches to the sequence starting at character number 12 just by looking at the locations of each k-mer, because like AAA starts at 12 and CCC starts at 24 and PTT starts at 36. So
excellent.
But these won't be coming together in any other place. We can read that.
Excellent. This is a very good observation. And basically, this is a paper we published in our group.
Okay.
So this is very good observation. This is one of the strategy to reduce the workload here. So rather than examining seeds individually, we could have some heuristics to link them together before that, check this location, and then we perform this computation. Yeah, I'm going to cover that as well. Yeah, question?
Sweet.
Oh, okay. Okay, perfect. Perfect. It's good that you observe that. This means you have the research mindset. All right, so I'm going now to cover the similarity measurement we perform at this step. So once we get the reference and we get the read, both of them of the same length now, and we want to check if they are similar or not. So we perform this using an algorithm, and I'm going to explain this algorithm next slide. Okay, we call this algorithm read alignment or edit distance computation. Edit distance is one example of this algorithm. And I will take it as an example, but there are many other algorithms as well. So this is just example. Edit distance, we define it as the minimum number of edits. By edit operation, we mean either you replace one character by another to make this sequence similar to the read, or you delete one character, or you insert another character to make this similar to that. You could take example Netherlands and Switzerland. So what kind of operation you can do to make them look alike. And based on those operation, you could infer how many differences are there between those two words. So we apply exactly the same thing between the read and the reference to know the number of differences. I have a couple of examples for you here. Imagine organization and operation. So we want to match them together. You can think about how many ways you could do that. So you have no restrictions about number of operations you could do or their location. So you could delete two characters here from organization, or you could insert two characters here in operation, for example. You could as well delete many characters from operation such that you make this looks exactly same as that. You could do it in another way. We have more examples, organization and translations. You could see how many ways you could do that. So you could replace here the O with the T, and then you insert some characters. You delete from here. You replace the Z with S, and so on. So how many edit operations you performed to make this look like that? This is basically the number of differences between the reference and the read. So how we do that in algorithm? Okay, basically we use dynamic programming approach. How many of you know the dynamic programming? Okay, perfect. Okay, this is a dynamic programming table where you have the reference segment here and the read segment here. Why we use dynamic programming, though it's computationally costly, right? Normally it's nested for loop. So it could be 2 loops, 2 for loops. So it could be quadratic time or more, right? Why we use that in the similarity measurement? So from the previous example, you could think why we need dynamic programming algorithm. Any hands? What we do here normally, we check if the first character match with the first. If not, we try with the deletion, then we try with insertion, then we try with substitution. Then we proceed to the next character. We check the first with the first 2 characters and so on. So this is basically what we are doing. So every time we pick one character from the first word, and then we enumerate all possible characters from the second word. And then we do the same thing with the second character, or combination of the first and the second character from the first word, and we enumerate all possible characters from the second word. And then we check which one give you the exact match with least number of operation, right? So you can imagine here O and O, we compare it many times, because we're trying to enumerate everything at every character of the first word. So we pick the first, then you enumerate first, 2, 3, 4, all kind of words. We have it from the second one, right? So we repeat the same operation over and over. And this is basically the idea of the dynamic programming. You perform the same computation over and over again, and you want to store it somewhere else and keep using that rather than performing it from scratch. So here, for example, when we compare the C, we store the value here. When we compare C and C together, so we store the value there, and so on. So whenever we need that information, we just go to that cell and retrieve it, then use it in the next cells, and so on. Okay, so this is the read alignment algorithm. We saw that it's built upon dynamic programming, and it's a quadratic time because we need for loop to maintain the table, rows, and columns. And because we use dynamic programming, we always rely on the previous cells. So imagine you are here. When you want to compute all the way from C to T, the similarity with C all the way to this A's, for example, to record it here or there, then you want to rely on the previous computation, right? So you cannot compute this cell before computing that cell, and that one as well, right? So this will restrict the way you are trying to compute the entire table. So either it will be row-wise, so you perform cell by cell in row shape, and then you proceed to the next row, because at this cell you rely on those cells, and so on. Or you could do it column-wise, so column by column, or you could do it anti-diagonal shape. So this will restrict the way you're trying to parallelize this algorithm. If you try to accelerate it in GPU, in processing in memory, or FPGA, or other accelerator platforms, you need to respect this data dependency in the dynamic programming. The other one is that we perform the entire computation towards the end until we know the exact number of differences. Then we decide whether we need to keep this segment or just eliminate it. And this is, again, costly. Imagine we have something really fast, not dynamic programming, something very linear, O(n) time, for example, can tell you whether this is correct or this is incorrect. This is a good match, or this is ingood or bad match. And this is one of the other work we have done in our research group, or we're co-authored with Professor Onur Mutlu. This is Mr. Fast, basically. This is a full read mapper that is built upon the hash table. So the problem here, we have a poor performance of existing read mapper because they are trying to check all the location in the hash table. So when we do breakdown to the execution time of the read mapper, we find that not more than 90% of the time is spent on the read alignment problem. So this dynamic programming, we use it more than 90% of the total time of the read mapper. So if we try to solve this problem, which is the large execution time of this particular part of the read mapper, then we could reduce the overall genome analysis time. So this is basically our goal in all the next works I'm going to present. So now let's revisit read mapping just to refresh about the process. So we have a read sequence, we get a k-mer, we check in the hash table if it exists or not. If it exists, then we got a list of locations in the reference genome. from the reference genome, we extract all those segments of the same length as the read we use. And then now we perform the read alignment. So we keep aligning one by one. This is a match. This is a good guy. Keep it. We align the next guy. There's a mismatch. We don't want it. Remove this guy. Remove that guy. And so on. So this is basically the original read mapper. So as some of you suggest that we could do some heuristics that could save this computation time. So we observed that most verification, as I mentioned in the example, one of them match and 5 of them mismatch. Then if we could find something to remove those mismatches in cheap or computationally cheap way, then we will save a lot of computation time. So we suggest to have a seed filtering. Seed filtering means that in the hash table, we don't need to check all the location that related to that seed. So this location list, if it has 1,000 locations, we need to verify only a few of them and remove all the rest. So this is what we call it seed filtering. The second way of accelerating such mapper, we call it pre-alignment filtering, which means after you extract the segment from the reference of the same length as the read, then you're trying to perform alignment. You stop at this moment. You don't do alignment. You bring a new algorithm, which is very fast. You examine if those two segments like having a good match or not. If potentially a good match, then you perform alignment. Otherwise, you just ignore it. Any questions for those two ideas so far? Is it clear? This is very important for the next work.
What is the complexity of that other algorithm that we are calling?
Okay, this is a very good question, and you can infer the requirement of such algorithm. So imagine if your alignment basically is O(n) or quadratic time, so at least you need less than that, right? If it is O(n), then it's perfect. But again, remember, you need to allow for differences. You cannot have something linear to check character by character and that's it. You need to allow differences, and those differences could be deletion, insertion, or substitution. So it's not just like skipping a character and move on. So this is, again, go back to our previous example. Now we will apply the two ideas, and we will see what will happen. So again, we will pick the read. We cut or chop it into k-mers. Again, there are many research on how to select your seeds. Then we check in the hash table. We get a bunch of locations. We go to the reference genome. Before that, we stop. We apply seed filtering. We try to remove them. One of the idea, he mentioned it, why not to check the adjacent location, right? If your seed located at this location and the next seed located at that location, so you could know that those two seeds next to each other appear in that reference, then yeah, this is potentially to be a good match. After removing or reducing the size of the location list, you extract them. Oops. Yeah, after you extract them from the reference, now you do pre-alignment filtering. You don't do alignment. This is not quadratic. This is very fast. You do, if it is potentially a good match, then you perform alignment. Then you proceed to the next. You verify it using your pre-alignment filter. You don't do alignment. If it is potentially not a match, then you just ignore it, eliminate it. We don't need the alignment in this case. So you can see both ideas reduce the workload, and both of them are necessary. All right, so we already checked the problem of read mapping, and now is the good time to go to algorithmic acceleration of read mapper. So basically, we will apply those two ideas I just mentioned, and we will see the exact algorithm, how it performs. All right, this is the first work that we published in our group as well. And the key observation is that adjacent seeds are normally located next to each other in the reference genome. So this is very simple heuristic or very simple observation. If you apply it, you will gain a huge benefit. So basically, we need to check the map location in the hash table. If it is next to each other based on the seeds, then this one a good guy. Otherwise, just eliminate it. Ignore the other locations. So this is the first key observation. The second key observation that this paper suggests is that always check the seeds that are cheap to compute or cheap to check. How we define cheapness here is basically if it is infrequent, then this is a good to check it. Why is that? Because if this seed does not happening frequently in the reference, then the location list will be small by itself. You don't need seed filtering here. So if it is infrequent, this maybe happen 5 times in the reference, then this is a good candidate to be examined. So this paper applies 2 ideas, adjacency filtering and cheap k-mer selection. The adjacency filtering, I have this example. This is a read, and you chop it into k-mer, for example, 3 k-mers, adjacent k-mers as well. And now, based on the location of each k-mer in the reference, you check whether those k-mers happening to be next to each other or far from each other. So which 3 k-mers you think that they are a good match?
The one on the far left.
Yeah, excellent. Okay.
But I also have one more question. I agree that it's a good idea to check if they are adjacent in the list. But even if they're not adjacent in the list, it doesn't really mean that we can just discard them, in my opinion, because we are expecting some sort of edit distance between our read and the reference genome anyways. And when we start to look at the adjacent regions now, the amount of the total bases we are observing are greater, so there's a greater chance that there's an error. And so if there's an error, there won't be a match, and then we'll say, okay, these don't match. But
all right, good question. However, this depends on the threshold you select. If you select 2 differences, and imagine the last seed is located, for example, 100 characters far from the previous k-mer, then definitely this is not a good match.
yeah.
So imagine this seed, those 2 seeds are located here, and this 3rd seed is located there. So the distance between the seeds is the main important thing here. So based on your threshold, which is you define it before you start, so you need to check based on that threshold whether the distance between the seeds is suitable or not. It could be single character, you could allow for that, still adjacent, right? But you don't allow for 100, for example, far, or 200 or 1,000. This doesn't make sense to have 2 seeds there and 1 seed there. Because at the end of the day, you will pick a segment of the reference that's equal to the length of the read, right? So you will ignore anyway that seed. And again, we don't have to chop the read into 3 k-mers. As one of you asked, if we pick them next to each other, or if we index the entire read, then we don't allow for differences, right? So basically here could be two k-mers, not 3. Or could be 1. Yeah.
But we could also be unlucky and have in every 3 of those small ones an error, and have a totally different mapping.
That's a very good question. I like it. This is basically based on probability that you will have the match with that read. And basically the sequencer machine try to sequence not single DNA, bunch of DNAs of the same sample. So you will have billions of those, though you need maybe millions of those reads to build the genome. Because of the read length 1,000, and you have millions of them, then you already satisfy the entire length. So for sure you have some extra, and you have some duplicates. And you have some repetition in the reference genome.
But you are interested in the difference from the reference genome to those which you read out by the machine, so the reduction in error from the machine doesn't help you.
Yeah, exactly. So when you build this one, it's not necessarily to cover everything in the human genome. So when you build it, after awards, you finish the read mapping, you will see there are some gaps because you ignore those reads. And this is somehow okay because we have like 25,000 genes. And normally when we do study, we are interested in one of those genes. We don't study all those genes together. So it's okay to have those gaps located like in other places.
So just to make sure I understand it correctly, if in this example here we had, for example, a T in the middle of the Cs, then we would not get the adjacency mapping, right? And we would not find this match, but that would just be a gap and improbable.
The thing, if you didn't have a seed in the middle, yeah, if you didn't have a seed here.
I mean one of the C's was just flipped and was a T.
Okay, yeah, if you have a seed here and that doesn't match with the reference, for example, and two of them match adjacent to each other, then this is fine. This is based on your filter, actually. You could specify those parameters if you want how many seeds, for example, in the read. If you have five seeds, for example, 1, 2, 3, 4, 5, and 3 of them exactly match, and they are adjacent to each other, then it's okay to tolerate that. Because basically you allow for 2 differences, so it's okay to have 2 k-mers mismatched.
Ah, okay, I get it.
That's why we don't rely on k-mers alone. There is a next step to align the entire read. So even the k-mer, basically the idea behind it is rather than, imagine you don't have a hash table. What's the brute force? To pick the read, to have the reference, check at this location, match, no, proceed next, match, no, proceed, proceed. You perform this OMN times. N is basically your reference length, M is the read length, for example. So you go character by character in the reference and you perform alignment. So the hash table is just to reduce this number of possibility. You don't want to visit every single character. So you have some of the seeds. That's why I don't want to go through the details. There are many papers mentioned how to select those seeds and which one to consider, whether 3 seeds is a good number, 4, 5, overlapped, non-overlapped, spaced, same length, different length. So there are many heuristics in this area. But it's good that you think in the next step. It's really good. I like your questions. All right, so we all agree that this guy potentially is good. So we need to perform alignment at this location. And we ignore those because, for example, this seed we picked from here, then the other seed is far away from the first seed. So it doesn't make sense to check seed by seed and perform alignment. So this is the same thing, and here how we apply it. We check this guy is 12 character long, the seed. We go to the seed location, is 12. This is the starting location. All of you agree that this is the starting location. So 12 plus 12, 24. So we are here at 24. Now we expect this seed to start at 24, right? Or close by, nearby that number, right? It depends on your threshold. So we find that the next seed that really exists at 24. So this is good indication. And now 24 plus 12, which is the length of that seed, then 36. Now we will check if the third k-mer here is located at 36 or not. We find it, yeah, it's there. Then yeah, this is a good guy. Go to the reference, extract the segment, and perform alignment or pre-alignment filtering. You go to the next location, 324 plus 12, 336. Does it exist here? No. So this is a bad guy, just eliminate him. You do the same thing for all those locations until you finish your hash table. Any question? Very simple, right? Okay, perfect. So now the second observation, we said that we want all the infrequent seeds. We don't want frequent one because the location list will be huge. We don't want that. So basically, how to know if it is frequent or infrequent, we have to go through the hash table one by one and check the location list size. So imagine this is your hash table, and for each seed, you have the location lists. So what you need basically just to count how many locations in each location list. So this seed has like five locations, this one 3, or this is 4, this is 2 locations. This is the number of locations basically. This is very huge, has thousands of locations. This is 2 locations, this is, I don't know, 2,000, 1 million. And you keep going through all the location lists. And then again, based on your threshold, you select which one you want to visit again and check each location of that list. So if your threshold, for example, 4, then this is good guy, good guy, good guy. Eliminate all the rest. And those are very expensive to examine because you need to go 1,000 times or less. So previously it was 3,000 locations to be examined, reduced all the way to just 8 locations. So you can imagine amount of time you save, because examining the hash table to check a word exists or not, it's all one, right? It's not expensive, right? But performing quadratic time algorithm, it's really expensive. So it's okay to increase the amount or the workload on the hash table rather than increasing the workload for the dynamic programming algorithm. Any questions so far? Okay, I'm going to mention the evaluation and take a break. So this is Mr. Fast, co-authored by Professor Onur Motlu. So they examine the performance on real data as well as simulated. And this is the evaluation setup. So basically, they could speed up the operation of the read mapper by those 2 ideas by 19x. This is end-to-end, the entire read mapper time. And this is basically the amount of workload reduced by using those 2 ideas. This is a logarithmic scale. So you could see that 99% of the workload is eliminated. So you save a huge amount of time doing that. So existing read mappers perform poorly in mapping billions of short reads to reference genome. Why is that? Because they don't apply any heuristics. They basically go and check all the locations. So most of the verification calculation are unnecessary. So you don't need actually to perform this amount of alignment every time. You could apply very simple heuristics and save this workload. And to reduce the cost of unnecessary verification, we apply seed filtering, pre-alignment filtering. In this work, only seed filtering. In the next work, we're going to see an example of pre-alignment filtering. And here where you can download the code, it's written in C. You could play with it, try to all the ideas you mentioned in this class. You have different number of seeds, adjacent by 1 character, 2 character, 5 characters. You pick, I don't know how many seeds per read, or you change the hash table. You make the seed length 10, 20, 30, and so on. So you could play with the code and check the performance. All right, this is a good stop point to have a break, and let's continue after the break. Thank you. All right, guys. So before the break, we were explaining about designing seed filtering algorithm to accelerate the process of the read mapper. And now we are going to present about the second idea of having a pre-alignment filtering. So all of us agree at this point that alignment is computationally expensive as we perform quadratic time dynamic programming algorithms. And we need to reduce the workload for that computationally expensive algorithm. So we need to optimize the operation of this alignment or this dynamic programming algorithm, or do some heuristics, some ideas, some fast algorithm to reduce the workload. So we presented the seed filtering again, and now we are going to present the idea of pre-alignment filtering. So by by moving the workload or the bottleneck from the alignment to a pre-alignment filtering, so the challenge now is to make the pre-alignment filtering really fast, much faster than the alignment itself, as well as make it accurate. So we don't want a pre-alignment filter to allow for the same workload to be moved from the pre-alignment filtering to the alignment. Otherwise, no benefit gained, right? So we want the pre-alignment filtering to be much faster than the alignment, as well as accurate in filtering. So this is basically our goal. All right, imagine you have a filter now, and what do you think the input of that filter will be? So this is a pre-alignment filter, basically. So when we apply this filter, so after, yeah?
After you found some matches in the reference.
Yeah, excellent. So we already have those seeds from the read. We check if they exist in the hash table. We get the location list, and we go to the reference, or we apply seed filtering first. Then we go to the reference, extract the segment. At this moment, we should apply our pre-alignment filtering. So just a moment before applying the alignment. So if this is the case, then the input should be a single segment coming from the reference and your read, right? And both of them of the same length. Okay, so when we should allow those two segments to pass the filter, and when do we need to eliminate them? Yeah? Go ahead.
When you found too many mismatches, then you...
Excellent. Okay, so if this is one of the properties of such filter. So when you have many mismatches, you don't allow those two segments to pass the filter, right? So we want to eliminate them. And when do we need to allow? When we don't have mismatches exceeding the user threshold. So basically, we need to filter out all incorrect mapping. We don't want to filter out any correct one. So all the correct ones, we want to preserve them. And we want high speed to gain the most benefit we can get out of pre-alignment filtering. Okay, so in summary, we need a minimal false accept rate. So we don't want to accept something that is incorrect. We want to accept all the correct one. So this is both of them lead to the same thing. So either you increase the accept rate, the reject rate, the correct reject rate. By this way, you minimize the false accept rate. And the second condition is that we don't want to reject something that is correct. We want to maintain or preserve all the correct ones. And we want as well to maintain the high speed. So the first example of pre-alignment filtering is SHD. Again, this work is co-authored by Professor Onur Mutlu. So in SHD, the key observation here, if two strings differ by E edits or E differences, then every base pair match can be aligned by at most 2 E shifts. And E here is the edit distance threshold that is set by the user. So what does mean this, this key observation? I'm going to explain it in an example in the next slide. So the key idea here is to do a bet vector to compute some computation there. Then you do logic operation on it such that you could achieve it in SIMD. The key result that SHD is 3x faster than the alignment and has only 7% false positive rate. False positive means that something shouldn't pass the filter, but this filter allows for it. So it basically rejects 93% of the incorrect ones and allow only for 7% of those to be aligned again by the alignment filter. So this is still good. All right. So to explain the key idea behind the shifted Hamming distance, I have this example for you. How many of you are aware about the Hamming distance? Okay, great. Hamming distance basically is the XOR all the corresponding characters, right? So basically, you want to know how many differences between this character and that, this and that, and so on. So when we do Hamming distance between those two words, Istanbul and Istanbul, the Hamming distance is?
0.
0. Okay, perfect. So we have exact match here, 8 matches and 0 mismatch. All right, we want to perform a difference. So ourselves, we want to tell that there's a difference here. So for me, I select to do a deletion. You could do insertion, you could do substitution, the way you want. So I will delete this character, then this word will be changed. So again, the edit is one deletion. This word becomes this one. And again, I want to perform Hamming distance between this and that. What do you think the result?
4. 5.
4 or 5?
Well, it depends how you define Hamming distance onto words of different length.
Okay, basically they should be in the same length, but imagine you have this one longer than that, then this one you could compare it with nothing or compare it with any character which is not similar to that one, then you'll get a mismatch, right? Anyway, here you'll get 5 mismatches, right? Which is wrong. Why? Because we only perform single difference, single deletion here at this location, right? So we cannot use Hamming distance in read alignment. You all agree on this, right? So this is the main idea behind shifted Hamming distance. So rather than doing pairwise comparison, which is the Hamming distance, let's do something with the word itself. So once we delete a character from here, we shift all the trailing characters to the left side, right? So if we try to shift it back to the right side, then we could get more matches. So let's try it. Now we have another instance of the same word, but shifted to the right direction, but by 1 step because our edit is single edit. If it is 2 edit, then we shift by 1 step, then 2 steps. Now let's do Hamming distance between the two words again. So you get 3 matches here and 4 matches here. So now how to combine the result of both copies?
Just addition.
Yeah, you can add this with that, right? Okay. There's one way to do that, but think about other ways, how you can do it in bitwise operation. Imagine you have another copies here, and you try to add all the matches coming from each copy.
Twice or.
Then you will have a lot of matches, right? And you may exceed, you may don't show any difference, right? You may end up having exact match with that. Imagine you have another copy here and you get 1 match or 2 match from that copy, then 2 plus 4 plus 3, then you're already in good shape, right? Okay, what do we do basically? We do XOR to do the pairwise comparison, which is Hamming distance. Then we convert this information into binary. So 0 for match, 1 for mismatch. And then what we do basically is just ANDing those 2 copies. By ANDing, you already have zeros, so you will cancel the ones here. You have zeros here, you will cancel the ones here, because 0 always dominates in the AND operation, right? And you end up having this 1, because here we have 1. 1 by 1 is 1. Then 1 indicates that there is a single edit at this location, right? Now your Hamming distance is correct, which we call it shifted Hamming distance. And this is basically the idea behind the shifted Hamming distance. It's just shift, XOR, and then AND operation, which you can perform it in FPGA very fast, GPU, SIMD, much faster than the alignment. There is no data dependencies here, everything pairwise. So you could do all of them in parallel, right? You could have this code and play with it again. You could optimize other ideas. You may not use AND. You may think about other ways of doing it. All right. So far we show that sequencing machine generate many reads, each of which potentially map to many locations, and we try to filter them using seed filtering. And then the filtering, the alignment itself, we try to accelerate it by using pre-alignment. However, the bottleneck now moved from alignment to pre-alignment filtering. So the pre-alignment filtering examined basically the entire workload and allow for some of it to go to the alignment, right? So if the pre-alignment is not fast enough, then we are adding more overhead. So now we are going to show a few examples about how to hardware accelerate such algorithms. Which is personally, I like to do this work. So if you are interested in any ideas accelerated in any hardware platform, just feel free to send email, meet in person, talk about it. All right, so again, here's one of the read alignment algorithm where you have the dynamic programming. Here's your reference segment, this is your read segment, and you try to fill this table. So based on each cell, you rely on previous computation. So we mentioned that there is data dependencies here. And this is the equation we follow up, how we fill each cell here. And this is what we are proposing. OK, same thing. You have the reference here, the read, and we are trying to fill it in different way such that we dissolve the data dependency. OK, what we do, just pairwise comparison, similar to the idea of Hamming distance and shifted Hamming distance. So A and C mismatch, so we record here a 1. A and T mismatch 1, A and A match 0. We continue all the way down until we finish the entire table. So here, what we have done is just removing the data dependencies. So no longer each cell depends on other cells, right? So you could compute everything in parallel, right? However, in this algorithm, the last cell will give you the number of edits, or you could do some step, we call it backtracking. I didn't show it here, but in backtracking step, you could directly get the number of edits in this two segments, between those two segments. Here we have only ones and zeros. So how to get some useful information out of this table? If you check the pattern here, you will see that there are diagonally consecutive zeros. Those zeros give some indications that there is a match between this portion all the way until this one. You see this zero, this, and this, and this, and that. This means it's perfect match all the way from here to T, and all the way from here to A, right? So if we could extract those zeros, and then we skip this one, and then we extract those zeros. Yeah, which are those zeros. So if we could find an algorithm to extract those zeros, then we already perform something much faster than the alignment itself, right? Then you could accelerate the entire operation using any accelerator you want. And this is what we have done in Gatekeeper. In Gatekeeper, we design a pre-alignment filtering. We implement it on FPGA such that we have the first FPGA-based pre-alignment filter. And this is your reads coming from the hash table. Then you decide on the location from the hash table. You extract the reference segment. Then before applying alignment, you perform pre-alignment filtering and then alignment. And this is Gatekeeper, basically. OK, so the first step in Gatekeeper is to generate those masks, the diagonal vectors. And here, I just rearrange the vectors such that we have them horizontally. So each diagonal vector is represented as horizontal line. So this was supposed to be a diagonal one, this is the other diagonal, and so on. So I just represent them horizontally to make it easier for you to visualize it. And now, where are the consecutive zeros? You can think about the longest one, right? We have one here, other, third, and maybe 4, right? So this is what we want, basically. If we could count the number of consecutive zeros here, then yeah, we could perform something very accurate and much faster than alignment. In Gatekeeper, we follow the same idea of SHD, such that we perform an AND operation between the vectors. So those zeros will cancel everything, those zeros will cancel everything, and so on. So here we have zeros and ones, and this will cause us a problem. What is it? We have two ones, and we want to keep them, because this means two differences, right? If we cancel them by AND operation, then we already remove the differences. So we don't maintain the first property of read mapper or read alignment. So we need to maintain those genetic differences. So if you perform AND operation, this zero will cancel this one, and this zero will cancel this one, then you end up having zeros here. So which is a perfect match. This one all the way once, so it's good. We don't have zeros. So the Gatekeeper applies the idea of canceling those short zeros. You have here two zeros and single zero that are not part of this consecutive zeros. So the key observation here, the matches are always long. The matches that are part of the correct alignment are always long, especially if you allow for a small number of differences. If you allow for one or two, imagine you have one difference here, then you have all these matches and all of these matches, right? If you allow for only one edit. If you allow for two edits, then you will have 2 or 3 long segments of zeros, right? If you have 4, you will have 5, which is E plus 1. Number of edits, or the edits threshold, plus 1, right? So if you allow for hundreds of differences, then you will have the matches very short. But normally we use less than 5%. The number of differences, less than 5%, especially if you compare human to human. So using this observation, we cancel all the zeros by flipping them. So any single zero or two zeros, flip them to ones. So this is before flipping, this is after flipping. You can see the effect of it. So here, no more zeros except that one. It's okay to allow for less differences, but not to cancel all of them. So certainly, those filters, it's okay to allow for some false positives, but it's not okay to cancel any correct one, okay? So now we do AND operation, and you get the result as this. Then you count the number of one, will give you the differences, okay? And the number of vectors here is basically 2e plus 1. So how we generate this one, for example, this one, Hamming mask, which is Hamming distance, just pairwise comparison. This one we shift to the right, then we compare. And this one we shift the read to the right by 2 steps, then we compare. This one we shift the read by 3 steps, then we compare. Why we do that? Because our edit distance threshold is 3, so we want to allow for 3 differences, and we don't know those differences are deletion or insertion. Could be 3 consecutive deletions. Then each deletion will cause the trailing characters to be shifted to one direction. So we need to shift 3 times to cancel the effect of the 3 deletions. Okay, so in this way we guarantee that to cover all the possible values or all the possible edits we could have. So imagine you have 1 insertion, 1 deletion, 1 substitution, or 2 insertion and 1 deletion. You could enumerate all of them and check whether it works or not. So after generating this, we count the number of ones, and this is your differences. And this is how to implement it in FPGA. Basically, you could flip those zeros in parallel. So this is one of the masks. So you have to go through the mask one by one and check 5 bits. Every 5 bits can be processed in parallel. Why 5, not 4, not 3? Because we always want to flip single zero or 2 zeros. Single zero mean 1, 0, 1, right? Otherwise, won't be single zero. So if 2 zeros, then 1, 0, 0, 1. So in this case, for example, we check 5 bits, and we flip the one in the middle. So that's why they are overlapped. So each, this is a lookup table, basically. Each 5 bits, we examine them, but we flip only the one in the middle. So we change it to here. So again, why 5 bits? So imagine this case where you have 2 zeros, and we want to flip both of them. So this one will be the target. So we need to examine to the left side 2 bits or to the right side 2 bits, because we don't know whether this one is 1001 or something else 1001, which is, this is the second case. So to cover both cases, we need to have 5 bits. Is it clear? So just only because of this case, because we are not sure whether the 10 is here or there, imagine if we have 4 bits, and the 4 bits started from here. So we are not sure whether the next one here is 0 or 1. If it is 0, it's not correct to flip it, right? Because in this case, we will have 3 zeros, not 2 zeros. And we want only to flip 2 zeros or 1 0. Okay? So again, similarly here, if the 4 bits are here, if this is your 4 bits here, so you are not sure if this is 0 or 1. So in total, you need always to check 5 bits of it in parallel and change the one in the middle. This is basically the most important part. And here how we implement it on FPGA. We have many processing cores in the FPGA board, and each processing core doing one alignment or one pre-alignment verification. So we transfer the read and the references from the host to the FPGA board, and each two or each pair go to single core. So you process here, verify, send back the result to the core, to the host, or you send it to the alignment directly to be verified if it is accepted. And all those cores work in parallel, and within the core, all those vectors are generated in parallel and processed in parallel, especially the flipping part, because all of them are independent of each other. And this is the chip layout after implementing the design. Those are some experimental results. And the important part of designing FPGA is that the design is limited to the data transfer from the host to the FPGA board. So it depends on the PCI Express you are using in the host, then you cannot satisfy more than the data throughput of that PCI Express. And for us, we are able to transfer 128 bits at this frequency. 128 bits means half of it has a read length. Why is that?
Because we need 2 bits to encode every base.
Perfect, exactly. So we have ACGTs, 4 characters, to represent them in binary so that we can transfer them to the FPGA board. We need 2 bits for each one, right? So if you transfer this much of bits, then you're transferring half of it as a read length. So if you want the double, then you need to wait double time to get the full read. Okay, so as you see in the chip layout, we are occupying only a small amount of the chip, right? And all of this is unutilized, right? So underutilizing a chip that costs $4,000 is not good practice as an engineer, right? So we are limited by the data transfer. If we could transfer more data, we could use this vacant area, right? So we could perform more operation in parallel, more pre-alignment filtering, right? So we could reduce the workload more and more rather than sending all of those to the alignment. Yeah, those the important results. The FPGA acceleration show 90x to 130x compared to the SSD, and 4x lower false accept rate than the adjacency filter, where you have the k-mers next to each other, and then, for example, you allow for one k-mer to be mismatched. So you have 4 k-mers exact match in this case. So end-to-end speed up, so if you add this pre-alignment filtering to a read mapper, you will end up resulting in 10x speed up. This is end-to-end. accelerating the entire read mapping. Again, a gatekeeper is available at this link. You could download the Verilog code. It's full design, so if you have VC709 FPGA board, you could burn the code directly and start using it, or you could synthesize the code and simulate it and play with it without the FPGA board. So FPGA-based pre-alignment greatly speed up the read mapping by 10x, as we show in the results. And FPGA-based pre-alignment filtering can be integrated with the sequencer because the sequencer has FPGA board inside it, which convert the chemical reaction or record it from this chemical reaction into a character, which is produced by the sequencer to the user in a form of file. We call it, FASTA file, or it could be the FASTQ files. It depends on the sequencer. So we could put the pre-alignment filtering inside the sequencer itself. So in the same FPGA board, we have a part of it utilized by the sequencer, and the other part is a pre-alignment filter. So the user, if he's a biologist, if he's a doctor, he doesn't need to deal with the FPGA board and do the pre-alignment filtering. So the sequencer machine will do everything for him. And this is the paper. And now the next idea is magnet. So we have the key observation is that those, if you still remember the consecutive zeros in each bit vector, are always non-overlapping. So one of them in this bit vector, the other one in the other one, the third one, fourth one, right? They are always non-overlapping. So based on this observation, why to do AND operation and mess up with the bits and try to flip some of them? Why not just go through each bit vector and count those consecutive zeros and then done? Then you could do add operation. So you could sum the length of all those non-overlapping consecutive zeros and infer the number of differences. And this is basically the key idea behind magnet. So we count the consecutive zeros in each mask and select the longest in a divide and conquer approach. How is that? I'm going to show here an example. So again, those are supposed to be diagonal bit vector, but I make them horizontally just to easily visualize them. So now those are our target. We don't know them in advance. So we have no idea where they are, the length of them. We have no idea of any information about those green segments. So basically, we go through each vector by vector and try to count the largest number of consecutive zeros. So we go through the first one. What's the largest number of consecutive zero? We find this guy. Then the second vector, we go through all the consecutive zeros. Here are 2 zeros. Here are another 2, 3 zeros. So this is the maximum. This is the largest. Again, same thing. So we find it, for example, here in this area. We go through all the vectors and select one largest per vector, and then we pick one globally largest out of all those largest. We collect them from the vector, which is basically, obviously, this one, right? This one should be the globally largest among all the consecutive zeros. So we pick this guy. And because this guy is shorter than the read length, the original read length, then definitely there is a difference at this side and this side. Otherwise, this match will be the entire vector, right? So for sure, there is a difference here and there. Then you also cancel this area, and you cancel +1 from right, +1 from left. In this way, we already canceled this longest consecutive of 0, and we store the length of it somewhere. We keep repeating this in divide and conquer fashion, which means the original problem was the entire vector. Now we take this as subproblem, and we divide the rest of the problem into 2 subproblems, one of them to the right side and one of them to the left side. So now this is a separate problem. This is another separate problem. So we repeat the same step, exactly same steps. We go through the first vector, count the consecutive zeros, nominate one of them as longest, then we go to second longest, third, fourth, then we pick one global longest out of all those nominated, and then again we cancel this area, we add +1 from here, and here the end of the vector, so we don't add anything. So we do this same thing for all sub-problems as well. Because they are independent from each other, then we perform those steps in parallel, because they are two separate sub-problems. So we repeat the same thing, the same steps, until we finish the whole vector. So now, where's the number of differences? So you have two options. Either to rely on the length of the segments you extracted, which they were green, or rely on the differences. It's up to you. If you sum up the length, the total length of all those green segments, then the same thing will give you the number of matches. If you know already the read length, so the read length minus those matches, you get the number of differences. Or you just simply count those yellow bars, which are the differences. Right? Easy and simple, very accurate, rather than allowing the way, or rather than clearing the way for those matches to appear in the AND vector, as we did in Gatekeeper and SHD. We're trying to remove all those obstacles, which are single 0, 2 zeros. We remove them so that the entire segment go up all the way to the result, right? But here, no, we go to that segment and extract it. We don't try to play with other bits. So this is a very accurate approach of counting the number of differences. This is how we do it in FPGA, just basically a counter that count all the consecutive zeros for each vector, and then you compare them, you pick the one out of those, and then you basically clear up this area where you find the segment, +1 from right, +1 from left, and then you keep repeating that. However, this is challenging to be implemented on FPGA. Why is that? Because of the nature of divide and conquer approach. So in FPGA, it's hardware. It's not C code. So you cannot have something random. As you don't know the size of the sub-problem, you don't know the location of it. It could be anywhere. It could be any length. So you cannot specify, for example, number of D flip-flops. How many D flip-flops you need? We don't know, right? So how we can change this code to fit in FPGA? Rather than dividing into two sub-problems here, you basically you need to flip all the bits, those to ones. Once you recognize this as the longest, so you flip everything within this rectangle or rounded rectangle, you flip it into ones. So in the next round, you apply the same algorithm, but to everything, not to this sub-problem and that one individually. You apply the same algorithm, but to everything, and because we flip all those to ones, so you won't get it in the next round, right? You won't recognize that as this as the longest segment of consecutive zeros, because it's already converted into ones.
But how do we recognize the differences then?
No, the differences before flipping. We already recognize this, and we add the differences, one from right, one from left, and basically you need to flip actually the column of the differences as well. You flip the differences and the segment this, and all the vectors as well that located within this rectangle.
Then you need to count the whole vector twice when you count the part where you already flipped again.
Exactly, you need to count it, but basically because you are counting the zeros, so once you find one, so you just skip.
Yeah, but isn't that
in FPGA, as long as you have enough of lookup table, so you could do that, right? You are not bounded by lookup table. At least at the design time, you already know that this vector is 100 long, for example. So you could have 100 D flip-flop for each vector, right? But if it is random, I have no idea how to design it, right? Unless you have state machine, for example, and you go bit by bit. And you try to do something, or maybe other idea is to restrict the search space rather than leave it as open as 100 character long. You could divide it into, for example, 10 rectangle, 20. You try to make it small as possible. Or you try to make some overlapped windows. You could think of other ways of doing that. So if you try to do one round per rectangle, so if you limit this search space into 4 portions, for example, and in each portion you do 1 round only, and you have enough overlapped, then you could achieve maybe same accuracy, maybe less accuracy, but you maintain high speed. Again, in pre-alignment filtering, speed and accuracy, both of them are really important. You could sacrifice a little bit of accuracy because we have alignment step in the next, which is very accurate. So you could allow for some false positives, but as long as the speed is important and fast, then it's still acceptable to have false positives. All right, so this is the idea behind magnet. You could see the differences between magnet and gatekeeper. So now the third idea is slider. I'm going to show the approach, and then we go back to the results. So this is again what we call it a neighborhood map, exactly same as the one we did in gatekeeper, where you have those diagonals and you do pairwise comparison between each character of the reference and the read. So this is how we compute it. And I highlight for you the consecutive zeros that are our target. Again, we have no idea about them in advance. So we don't know where they are, what's the length or the order. We don't know anything about it. And I could do it horizontally to visualize it, but this time I want to do it in this way, just to remember how we transfer the read alignment to pre-alignment filtering. So this is exactly as we did in alignment, but we don't have data dependency here again. So just the same problem, we need to collect those consecutive zeros, the way you like. Think about it in a different way. You may have a new idea. You may implement it in your own and see and prove it's working fine. Then you can proceed to accelerate it in GPU, FPGA. Okay, in this algorithm, what we do, we have now you could have or you can see the reason why it was challenging to implement it in FPGA. Now we limit the search space rather than searching the entire vector. So we limit it to this window. So this window is basically 4 columns. So it's fixed to 4 columns, regardless of the number of rows. So we always want 4 columns to be in our search space. And we go diagonally, and we search the number of zeros within each vector. So this is the first window, 4 columns. And now we want to track the diagonal matches. We keep diagonally counting one by one how many zeros we have. We find this guy has 4 zeros, which is potentially our target. So we will pick this guy now. We store it somewhere that we have 4 zeros at this location from here to there. And now, because we will do 1 round within a window, so it won't be accurate if we have one, for example, here, and we have 2 zeros. So we need to search at every column. So at every column, we keep sliding this window. So now we will slide the window by 1 column. Again, the window size is fixed to 4 columns. We keep repeating the same thing, and we find this guy has 3 zeros. So we pick this guy. And now when we move it to the what we call it slider bit vector, because we select overlapped window, so we will have a conflict at those 3 bits, right? Because they were previously coming from the first window, and the second window again producing results. So we need to break the ties between those 2 windows. Which one to select? The zeros coming from the first window or the second window? in case of zeros, it's not a problem, right? All of them zeros, then fine. Same value, right? Doesn't matter where it does come from. But in case we have 1 and 0, 1 coming from second window and 0 coming from the first window, what we should do? Okay, to be on safe side, we don't want to reject any correct mapping. Okay, so we always want to maximize the number of zeros, and we want to minimize the number of ones. So just to be on safe side, there is nothing related to biological reason here, right? So just to not cancel or not eliminate any correct mapping, we want always to maximize number of zeros. So if this window give us 3 zeros and 1, single 1, and the previous window give us 4 zeros, for example, then we will have the zeros instead of the 1, just to maximize the number of zeros. This is how we break the ties between the window. Otherwise, we just proceed exactly same steps, and this is what we get from all the windows all the way up to here, which is this window. And now easily you can count the number of ones here, which will give you the number of differences. Again, this one is perfect fit for FPGA implementation. Why is that? Because the search space is fixed. So we have 4 columns, so we need to have 4 D flip-flops per row. But again, to count using D flip-flops, we need 4 clock edges to count, right? So at each clock edge, we count, right? Until we have the number of bits for word of length 4 bits, right? So you could do it in lookup table because it's limited to 4 bits. So the number of possible values, you could enumerate it to 16 values, right? So you could search using lookup table rather than waiting for 4 clock edges, right? So in this way, yeah, here are the 16 values we have. So you could search if each vector has this value, then exactly as we do in hash table, if it has this value, then the number of zeros is 2, here 3, 2, 3, 3, 4. And one thing to note here, that we don't count consecutively. We just count the number of zeros. Again, this is another reason that we want to maximize the number of zeros. So we don't care about whether being consecutively or not, because they are overlapped. So for sure, we will have those consecutive zeros, which are our target, captured by the window and recorded in the slider bit vector. And here we have, for each bit vector, this counter. Again, this counter implemented as lookup table. So in single clock edge, you could have the results ready for you. And again, all those search windows are independent of each other. They are not dependent on the data coming from the previous window. So you could perform all those search operation in a parallel. But when you come up to the slider bit vector, you want to store those zeros, right? So you need to wait for the result to be read from all of them. Then you can decide whether I will take the result of that window or this window and so on. Okay? So in the FPGA, we have always all lookup table. All slices in the FPGA produce the result in the same time, regardless the operation you do. If you do single AND operation in the lookup table, or if you do tens of them, as long as it fits to a single slice or single lookup table, then the result will be produced in the same time. So you don't have to worry whether those windows will produce the result in the same time or not. So one important result between slider and magnet, where you search the entire bit vector, magnet occupies 10x the resources needed for slider, for example. So you can definitely infer the reason.
And you said, like, for the slider, the one before limitation was the PCA bandwidth.
Yeah, PCI Express is still a bottleneck here.
But your card, which is display, is like only like 4x Or aren't there like FPGA with 16x bandwidth?
Sorry, what?
I mean, you said the PCA is the bottleneck, but I don't know if it's the card you're using or not, but there's like 16x PCA or 4x PCA. And so there are no FPGA with 16x, or is it just picture you have?
This one, the VC709, Vertex 7, it uses PCI Express 3 with 8x. So in this one is limited to 13.3 gigabit per second, or double that number, which is basically it uses frequency of 250 megahertz. You are allowed to transfer 128 bits, or up all the way to 256 within that frequency. So this is the bandwidth. This is how you specify it. So it will be like 5 nanoseconds. So every 5 nanoseconds you transfer 128 if you use 4x. If you use 8x, then you will transfer 256 for that same amount of time. So this is your bandwidth. So as long as you have much data to process, then you could increase more cores in the FPGA to do more things in parallel. But again, I'm out of time. But yeah, if you increase the accuracy of the filter, even if you're still limited with the same bottleneck, so gatekeeper, magnet, slider, same bottleneck, data transfer, we cannot achieve more. But the difference between those three filters is the accuracy. So if you achieve highly accurate algorithm, then with the same bandwidth that you transfer to gatekeeper, for example, you could achieve, you could produce less workload to the alignment. So this is how you end up saving more time, though you are still bounded by the data movement. So if you increase the accuracy of the filter, you reduce the workload for the next step. This is basically the main idea. All right, so let's proceed to the processing and memory. So again, we are bounded by the data movement from the host to the FPGA to the GPU, from the hard drive to the memory or to the processor. So still we have a huge amount of data to move it from here to there, back and forth, right? Because we pick data, process it, and then store the result again into another file, right? So a smart idea of doing this, why not to process it in memory or near cache, or do something to save the journey of moving this data to the processor back and forth. So here's the contribution of this paper, again, developed in our lab, is that we want to do a processing in memory to alleviate the bottleneck of data movement. So we don't want to optimize the operation as much as we want to reduce the data movement or speed up the process of moving the data. So we need to design mapping and filtering algorithm that fit processing and memory. So the first step, we want to build our own data structure. No more hash tables. We don't want that. We want something that enables us to process while the data in memory. So, and this is the idea behind the bins. We have this reference genome. We split it into overlapped bins. For each bin, we capture, for example, 100 characters. And then we enumerate all possible values of a word that has 5 characters. So all A's, then A's, C's, A's, T, and so on. We enumerate all possible values for that word. And then if this word happened in that bin, then we record 1. If it doesn't exist, we record 0 for that particular bin. If we have another bin, we keep the same steps. We repeat it, but different result, right? Because different content. So we do all of them to all bins. And we have this result. So now we have the read. That, by the way, that step, we perform it only once. Sorry. Yeah, this step, we perform it only once, which is for the reference, exactly as we do with the hash table. So now the time to check if this read exists in, let's call it hash table or our own bins, right? So for this is a read, and we pick a bin of same link as the one we created in the reference genome, exactly as we do with the seeds. And we enumerate again exactly the same number of possible values for a word of length 5. And we check if this exists, if this word exists in the bin we created previously for the reference or not. So we go through each bin by bin, and we check if it exists or not. It exists here, so this is 1. Exists there, then 1. Exists here, 1, 1. Otherwise, it's zeros, right? So this bin is created for the read. Now we compare this bin with the one we created for the reference. Is it clear so far? It's very straightforward. So basically, as we did in hash table, we have seeds, and we try to check if it exists or not exists. So those all the bins coming from the reference. And now we want to check if a read exists or not. So we do exactly same steps. We have overlapped tokens or seeds from the read, and we check if they exist here or not. So we have now 3 tokens, and if you sum up the results, which is exist or not exist, then 1 from here plus 0 plus 0 plus 1 plus 1 and so on. So you sum up those results, which will tell you how many of those seeds exist, basically. Which is a way of comparing the bins together. And is it more than the threshold or not? So exactly same idea with the k-mers, but without the use of hash table. In this way, it enables us to use it processing in memory. So we have here, for example, 1, 2, 3. We have 3 seeds match between the read and this bin. So same idea. If it is exceeding the threshold, then just eliminate it. If it is still respecting the threshold, so just maintain it. And now how we do it in full mapper setup. So you have the read sequence. You have all those seed locations. So now the things, so you have this location, whether it exists or not, in the bin. So now we will sum up those results, and we'll tell you which one to keep, which one to discard, if it is exceeding the threshold or not. So if this is fine, you send it to the reference, you extract from the reference the segment you like at that location, and then you send it to the read alignment or the distance calculation. Okay, so far nothing new except the way we implement it. However, for the next steps, I'm going to show you the implementation in memory. So in 3D stacks memory, you have DRAM layers and you have a logic layer where you could perform some computation. So it has extremely high bandwidth, as well as stack customizable logic layer, where you can perform simple logical operations. So we want to implement the entire Grim filter inside the memory. So if your data is ready here, we want to exploit this logic layer to do some operations. And as you can see, the bins, you could perform it in parallel, and you could use bitwise logic operation, right? Because it's just you check if it exists or not, and basically you sum all the results, and you check it with the threshold if it is exceeding or not. So that's all, right? So this is for Micron HMC architecture, and this is how you implement the bins. So each layer is organized as an array of banks, and each bank is an array of cells with a row buffer to transfer the data. So you need to access this row buffer before you transfer the data, basically. So you can read rows at the same time. So in this case, because the bins are independent of each other, so what we want to do basically is the first bit of each bin to be in different layer. So those bins will be in vertical. So when you read, you read them in parallel. All of them, you move them to the row buffer. And you keep adding the operation one by one. So in this way, you end up processing all of them at the same time. You don't go bin by bin, but you process all of them in parallel. So this is how you access the bins. You move them to the logic layer, perform operations. We have about 10 minutes. So those details about the implementation, and here is the great side of accelerating processing and memory. So the two important performance metric here is again the speed and the false positive rate, or they call it here false negative rate. False positive rate means something not good and you're allowed to pass. This is okay, fine, because we have another step that very accurate, so it can fine-tuning this stuff. But to have false negative means something correct and you delete it. We don't want that. We don't want to delete the correct ones. Otherwise, we won't get our constructed reference genome. All right. So this is the results. So Grim filter, which is a filter implemented in a memory, could achieve 1.8x to 3.7x benefits for all real data set with 2.1 average performance benefits. So this is fast hash filter, the one implemented. Implements the two ideas I mentioned, which is adjacency filter and infrequencies. So this Gram filters compare the result with the fast hash. The false negative here, it reduced, actually in this slide, the false negative, it's looked at it as false positive. So you can think of it here as a false positive. It actually depends on the way you are looking for it. If you are at the alignment side and you're looking at a false positive, which are incorrect coming to you, so this is a false negative, right? But if you are at the pre-alignment filter side, then this is something incorrect and coming out of you, then this is good, actually, because there are next step there, so it can filter out those. So you can think about it here as a false positive. I should change it, actually, here. So this is more accurate than the ideas presented before, because it shows 5.6x to 6.4x reduction, which is something good. It's better to reduce the false positive. So this is the paper. You can have a look at it and check it if you're interested to improve something in it. Check, for example, you want spaced bands, not overlap, non-overlap, different length. you could always discover different methodology how to do stuff, because this area is recent, start to appear using the pre-alignment filtering just in the last 4 or 5 years. So whatever you change with the algorithm, it will be something new. But it should be implemented in a clever way, in a smart way, where you utilize your resources efficiently. You don't want to use an FPGA for just single core. You just do single comparison, because you will pay a cost for moving the data from the host to the FPGA. So it will be more wise to align or to check those segments in the CPU rather than moving it to FPGA. Although the FPGA is fast, but only to transfer single segment doesn't worth it, right? Because you will wait for the data movement from the host to the FPGA. So you need always to perform like hundreds of those segments in parallel in FPGA to gain this much of benefits. So your algorithm, basically, you need to be very fast, occupying a less amount of resources such that you enable processing more and more in parallel. And you need basically to have it accurate. So by combining all those parameters, so you could have the best pre-alignment filter so far. And if you are able to achieve 100% accuracy, then this is read alignment. Then you remove the dynamic programming and you have your own algorithm, which I believe will cause a revolution in this area, because everything is still depending on the read alignment, which is the last step. If you could have ON read alignment, so this is something great. So the future direction, and personally I'm interested in developing more read mappers that work for both short and long reads. So we want something, we prefer always short reads because they are accurate, but the accuracy of long reads as well is dropping down very fast. For the last years, we can see the size and the cost of those sequencer is really improving very fast. So if you design something for the future, though the long reads are still like limited, the use of them. So if you design something for the future, it will be great for the time being, because by finishing this design by 2-3 years, you will have this long reads very accurate. Then you could use your own design. And basically, I'm interested in more pre-alignment filtering. If you have ideas, if you want to share with me, you want to develop some ideas together, we could work on it. We could accelerate it in other platforms to achieve more speed up. Something that fit for FPGA maybe is not good for GPU, something good for GPU maybe is not good for processing in memory, and so on. So you really need to target the accelerator carefully. This is some slides about the 3rd generation sequencing technology, which generate the long pieces, which are easier to link together because you could link 5 pieces of them, then you already have the sequence, rather than linking like billions or millions of those small pieces. But again, the issue here is the accuracy. So we have those devices currently. This is available in the market, which is a USB stick. You could put a sample and you analyze it, you get the sequence out of it, or the reads. And again, we have iPhone gadgets as well. This is the technology how we sequence the long reads, rather than chopping into short reads randomly. So start to read the sequence. But again, we don't need labeling of DNA nucleotides, so we don't need to convert it. So that's why we are very fast, and we could allow for a very long reads. But it relies on electronic or chemical structure of different nucleotide for identification. And yeah, and this is the pipeline, how we do genome analysis for long reads. Basically, here you find overlaps between those reads. using some other algorithm than the one we presented, and then we do assembly, so you try to link those three together. Then you try to do mapping with other reference genome. Then you do polishing. Polishing is something to reduce the errors in the long reads. So there are some heuristics to reduce those using other information coming, for example, you already mapped to a human, so you have some short reads for another human, so you could use them, or for the same human, so you could combine long reads and short reads. You align them to the same reference, and based on the overlap between the short reads and the long reads at the same location in the reference, you could probabilistically say, okay, short read is more accurate and the character was A at this location, so let's change the character in the long read to A, for example. So this one, a lot of heuristics there, and Jan Fortina sitting in the back is working on this area on improving the accuracy of long reads assembly. So you could talk to him as well on developing some polishing heuristics, some algorithm to improve the accuracy of long reads. So if we could solve this issue, then it will be great. Then no need to spend more time on short reads. And this is a paper presented by our group about the long read sequencing technologies and what are the existing algorithm, which one to use, which one is better than the others, advantage, pros and cons, and so on. So I'm going to conclude system design for bioinformatics is a critical problem because, again, it has implication on clinical aspects and our persons and personal issues, large scientific discoveries as well to discover which gene causes which disease, and then you do personalized medicine for that disease based on the gene. If you have a way of altering that gene, then you could terminate that gene from causing that disease, for example, which is, we call it a methylation. So this talk, we talk about how we do algorithmic improvement, how to accelerate the entire read mapping, and how to accelerate it in hardware as well, FPGA and processing and memory. We talk about read mapping process, which is the bottleneck in genome analysis step. And we presented several ideas that are homegrown in our lab. And we are still looking for more ideas to develop a complete system to do read mapping. We don't want just basically to have those individual steps. We want to combine them all together and have our own system that is connected to the USB stack sequencer, or it could be Apple Watch, something like for future design. And we have our own read mapper connected next to the sequencer. So you could do both of them with very small device at low cost, high speed, and high accuracy. This is basically our objective or our ultimate goal. So I'd like to acknowledge Prof. Onur Mutlu, Prof. Can Alakan, and many students from our group working on all of those papers and those topics. Thank you so much for paying attention and having all those great questions. I can see you're already interested in many aspects of this area. So again, if you want to develop some idea, you don't know, you could think about it as simple, but it could change a lot in the algorithm, the speed, the design, and etc. As you saw, most of those ideas really simple, but very efficient. Thank you.